to talk about the unintended consequences of single-base DNA editing. So as Jenny just covered, single-base DNA editing usually uses either cytosine base editors, which we're going to call CBEs, to convert CG base pairs to AT base pairs, or adenine base editors, or ABEs, to convert AT base pairs to CG pairs. So while these base editors are used for targeted nucleotide alterations in DNA, they've also been shown to edit RNA in an effective cell and produce off-target DNA and RNA edits. So this first thing that we're going to look at is the off-target DNA edits. So why do we want to look at these off-target DNA edits? So we use these base editors, or we hope to use these base editors for research and for therapeutic use. However, they might have limitations if we have off-target effects that produce consequences that we can't predict or control. So this study, cytosine but not adenosine base editors induce genome-wide off-target mutations in rice, studies the off-target DNA editing by CBEs and ABEs. So most existing studies use in silico programs to predict probable off-target mutation sites and analyze only those sites or sites proximal to the target sequence for these types of mutations. So this study seeks to use whole genome sequencing to provide an unbiased and more complete picture of the off-target effects of single base editors in DNA. So this study compares the effects of BE3, HF1 BE3, and ABE base editors. So BE3 and HF1 BE3, which is also known as high fidelity BE3, those first two are CBEs, or cytosine base editors. ABE is an adenine base editor. So as we can see, we have a layout of 6 different genetic units on this side of the screen. So the first that we see laid out is BE3 and HF1 BE3. So these are remarkably similar. The only difference between them is that the nickase Cas9, which Jenny discussed a little bit earlier, is high fidelity in the high fidelity BE3. As we move down, though, we can see that the ABE exhibits differences in its structure from the BE3 and the HF1 BE3. So to have a base editor, all of these different types, both the cytosine base editors and the adenine base editors, need to have a nickase Cas9 unit, and they must also have a deaminase unit. So in the cytosine base editors, the deaminase unit is called R-ApoB-C1. So this is rat ApoB-C1, which catalyzes the deamination of cytosine. In ABE, we want to degrade adenine. So instead of using the R-ApoB-C, we'll use something called EC-TAD-A, which is a deaminase for adenine. So the last notable difference between these first three genetic setups that we see is that both of the CBEs have this unit UGI. So that's also something that Jenny talked about when we were first comparing these. The UGI is the uracil glycosylase inhibitor. So that is notable in that it's present in the CBEs, but not the ABE. There are also 3 DNA units that are shown on the bottom of the diagram. So these are the exact same ones as the BE3, high fidelity BE3 and ABE that are shown on the top of the diagram. However, these lack the sgRNA portion seen on the right-hand side. So without this sgRNA portion, these 3 sections down here are not actually targeting any kind of specific sequence in the DNA. So this will become important when we see the comparison between how the CBEs and ABEs that have a guide RNA and thus the target sequence affect the off-target DNA effects versus these base editors that do not contain a guide RNA sequence. So the way that this study was set up is that rice seeds were transformed using plasmids that contained one kind or none of these gene units that were on plasmids. So these rice seeds were transformed, and then the transformed cells were allowed to grow for 11 to 12 weeks into plants. So the base editor plants that they have had BE3 with and without the single guide RNA, and they had high fidelity BE3 with and without the guide RNA, as well as ABE with and without the guide RNA. They also had many control plants that were not transformed with anything. So once they had all of these different types of plants, they wanted to see the different effects in off-target mutations that they could find within the whole genome and compare the levels that were induced by these different types of base editors. So to do this in a more complete way than had been done before, rather than looking for predictable sites, I think the technology that they had used is something called Cas-off Finder, which is able to predict probable sites where mutations might occur after being treated with a base editor. Instead of doing that, they used whole genome sequencing, and then they used two different types of programs to analyze the whole genome sequencing results. The first one is GATK low frequency Strelka2, which checks for single nucleotide variants, or SNVs. So that would be any type of change that was catalyzed by these base editors, because it would look for a single base pair that was different than it was before. And then there is also the program GATK Strelka2 that searches for indels, which are mutations that are insertion or deletions. So this would mean there's an extra DNA base pair where it shouldn't be, or we're missing one where it should be. All right. So they compared the results of this whole genome sequencing between the plants that were transformed with different types of base editors and the ones that were just controls. So in this whole chart on the right-hand side, anything that is noted by orange is going to be a control. Anything that's noted by green is going to be transformed by BE3. Anything that's blue is transformed by high fidelity BE3. And everything that's red is transformed by the ABE. So these charts show the results that were found in whole genome sequencing that reference the number of indels or insertion deletions between these four different groups of plants. And then they show the frequency of different types of single nucleotide variants that were in between all of these plants. So looking at part A, we can see that levels of insertion deletion mutations in between the control, the BE3, the high fidelity, and the ABE are all relatively similar. So this suggests that the addition of base editors has almost no effect on the number of insertion or deletion mutations that you can find in the genome. If we move over, though, to part B, we can see that the amount of BE3 and high fidelity BE3 mutations that are single nucleotide variants are much higher than the ones in the control and the ABE, which are about the same frequency as each other. So this suggests that introducing a type of cytosine base editor has more off-target effects with regards to single nucleotide variant mutations. The next thing that they wanted to identify is what type of single nucleotide variant mutations these are. So to do so, if we look at part C of this diagram, we can see they rank the frequencies of different types of single nucleotide variants throughout the different transformed plants. So the ones that are very low to the ground suggests that there's very little incident of single nucleotide variants, or that they're very similar to normal levels in untransformed plants. But if we look at the C to T and G to A transformation that's listed on the right side of the graph, we can see that the green and blue, which are the BE3 and the high fidelity BE3, plants exhibit a higher number of C to T transformations than the control and the adenine base pair editors do. So this suggests that, as we expect, since these BE3 and high fidelity BE3 are both cytosine base editors, they produce more cytosine to thymine edits. However, they're doing it more frequently than they should be. I just said that wrong. The BE3 and HF1 BE3 editors are causing a higher number of SNVs that are C to T transformations than the other versions. So this is confirmed when we look at part D, where it shows the number of SNVs that are C to T transformations, which is what the cytosine base editors BE3 and HF1 BE3 catalyze. And as expected, we can see that the BE3 and the HF1 BE3 plants exhibit much higher rates of these SNVs that are C to T transformations than the control and the ABE. So what's interesting, though, is that if we look then at the part G of this table, we can see the number of A to G single nucleotide variant mutations that are present in all 4 plant types. So since we have for the cytosine base editors a higher number of C to T SNVs, we would expect somewhat to see that the adenine base editor also exhibits a higher A to G SNV, since that's what they tend to catalyze. But in looking at G, we can see that the number of A to G single nucleotide variants is actually stable across all 4 plant groups, with all of them matching the level of variance as the control. So this suggests that the ABE editor does not actually produce many A to G single nucleotide variants in DNA. So the next thing that they wanted to look at was the genomic distribution of these C to T SNVs. So if we take a look first at this round chart in the middle, this shows us the distribution of the C to T SNVs throughout different chromosomes on this bacterial genome. So if we look at this, we can see that each color band corresponds to a specific number of C to T transformations in each type of plant. So if we look at these bands of color correspond to a differently transformed rice plant. So we have the control C to T SNVs, and then we have the rate of C to T conversion for the plants that are transformed with the adenine base editors and the cytosine base editors, both with and without single guide RNAs. So as we can see through here, as we go around this genome through all of these chromosomes, the color of each band, or like the density of each band, stays pretty much the same as we go our way around. So this suggests that the number of single nucleotide variants is pretty much evenly distributed throughout the whole genome. The next thing that we can look at this graph is the number of C to T single nucleotide variants in the genic regions versus in the whole genome. So we can see that these variations are higher in the BE3 and the high fidelity BE3 transformed plants, which is also confirmed in part C, where we can see that there's a much higher rate of conversion in the intergenic regions versus any of the other regions in the whole genome. So from these charts, we can determine that BE3 and high fidelity BE3, which are both cytosine base editors, induce genome-wide mutations in rice. However, the ABE base editors do not induce mutations in the DNA of rice, as far as we can tell, or at as high of a level. So their off-target mutations were mainly C to T single nucleotide variants, which is consistent with their function in that they tend to replace CG pairings with TA pairings. And we can see through the analysis of this last slide that these mutations are pretty much located throughout the entirety of the genome. So this research was very helpful in providing a more complete picture of what the off-target effects of base editors are in DNA, because as we can see, since the distribution of these mutations is very wide and spread out through the genome, it can't be predicted by current in silico approaches, which only use specific programs to analyze where probable mutation sites are. If we were only to analyze probable mutation sites, there's a big chance that we couldn't find many of these variations that we see through this whole genome sequencing. So to minimize off-target base mutations, we would need to have optimization of the cytidine deaminase or the UGI components, because those are the ones that are different than the ABE ones. And the ABE does not exert nearly as much DNA off-target deamination. All right, so the next thing that we're going to talk about is another unintended effect of these base editors. So these base editors not only produce unintended effects in DNA, but they can also produce unintended effects in RNA. So CVEs have been already shown, as we just went over, to produce off-target DNA edits, but there's been little research into the RNA editing effects. So new technologies of single base editors are very useful in research and therapeutics, but we need to understand any unintended consequences that may lead to negative effects. So for this paper, that's based on transcriptome-wide off-target RNA editing induced by CRISPR-guided DNA base editors, in order to try and diagnose the off-target effects of these editing tools in RNA, the transcriptome and genome of liver and kidney cells were analyzed to find the amount, location, and type of off-target edits due to the introduction of CVEs. And then CVEs were introduced in different concentrations and with various mutations to determine what part of the CVE is causing these off-target effects. Throughout this paper, they did find that both CVEs and ABEs cause numerous off-target RNA edits that have no association to DNA edits. This is important because we want to make sure that these edits we're seeing are made directly to the RNA and are not just unintentional DNA edits that were transcribed to make mistakes in these RNAs. The RNA edit locations don't correspond to any specific guide RNA targets, and almost all of the CBE mutations were CDU changes. So the off-target effects of CBEs and ABEs are also shown in this paper to be able to be reduced through modification of the deaminase enzyme. So let's get into it. All right. So the first thing that they looked at was RNA editing that's induced by BE3, which is a cytosine deaminase base editor, in HepG2 cells, which are human liver tissue cells. Okay, so RNA editing induced by BE3 and HepG2 cells. So HepG2 cells are liver tissue cells. And for the purpose of this study, we're going to look at the effects of BE3, which is a cytidine base editor. We're going to look at the effects of that on RNA edits in HepG2 cells. So this CRISPR-Cas base editing technology enables targeted nucleotide alterations. And the most widely used cytosine base editors induce deamination of DNA cytosines using something called rat ApoB-C enzyme. We can see that right here on this chart. And this is linked by a Cas protein RNA complex. So overexpression of ApoB-C can lead to modification of cytosines in many transcripts other than ApoB, which is its target, and in multiple cell types. The HepG2 cells that we're going to talk about right now were transfected with plasmids that had either BE3 or a negative control fusion. This negative control has all of the same parts as BE3, but it doesn't contain our ApoB-C1. So the purpose of trying to figure this out is seeing if the R-ApoB-C1 has any effect on whether or not there are changes that are unintended to the RNA produced. So we tried to transform these cells using both the positive with R-ApoB-CB3 and the negative control with BA3 minus the R-ApoB-C, along with a guide RNA that was targeted to a specific site in the genome called human RNF2 gene site. So the first thing that we want to see is if this BE3 is able to target the RNF2 site and produce edits there efficiently. So in order to test this, if we look at B on this side, we have the BE3 on top represented, and we have the control on bottom that's BE3 minus the R-ApoB-C. So as you can see, these areas with the purple represent areas where the C to U transition took place. So we can see a high amount of purple in both of the C targets within this target DNA site when we're looking at the regular BE3. However, when we're looking at the control, there is almost none. So this can show us that, yes, the BE3 with the rat ApoB-C1 is able to efficiently edit DNA at its target site and produces the intended effects where we have the C to T deamination. The next thing that happened was that RNA editing was assessed using targeted RNA amplicon sequencing to look at the cytosines in the ApoB transcript. And these results showed that there has been many of these RNA cytosines were transformed due to this CBE, both with and outside of the DNA target sequence. So transcriptome-wide RNA editing with BE3 was assessed using RNA-seq of the entire RNA. And it was found that RNA base positions that were altered in cells expressed BE3, but not control cells that expressed the BE3 complex lacking ApoB-C1. So the vast majority of these changes were C to U distributed without the transcriptome. So if we look at this part in C, we can see that the first column is labeled GEP or GFP. This is for the negative control. So this one right here with the GFP was transformed to the plasmid that contained the control sequence, which is B3 minus the rat ApoB-C. And we can see that the RNA C-to-U editing is very low. These 4 columns on the right are 4 different trials of the same conversion where the organism was transformed with BE3 that did contain R-ApoB-C1. And in all 4 of these replicated experiments, we get very high C-to-U editing. And if we look on D, we can see that this editing is very well spread out throughout the entire chromosome. It's also shown on E that in all 4 of the replicates, which have DNA editing for C to T induced by the BE3, almost like between 42 and 45% of expressed genes had 1 or more edited C, which is a whole lot. The next thing that they tried to find out was whether there was any kind of similarities in the places where these base editors were binding, even though they weren't on the target sequence. And what they found in all of these replications is that they tended to bind to a consensus motif that was A, C, and then had its third site as usually a T or an A. So this made sense to them because AC, and they call it W, which would represent T or A, is a normal target site of wild type rat ApoB-C1. So that would make sense as to why these sequences are targeted in the cytosine base editor that encodes our ApoB-C1. In order to show that these edits are made in all types of cells and not just in one specific type of liver cell tissue. They replicated the experiment in kidney cells, and it yielded similar results, confirming the fact that the CBE with rat ApoB but not without it was able to produce large effects in RNA CTU editing that were not expected. So obviously this presents a problem. We don't want to have unintended consequences for our editing. The whole point of having this kind of specific targeted single base editing is that we're able to change very specific parts of the genome. So having large off-target effects is not exactly ideal. So during the next part of this study, they tried to find B3 variants that had reduced RNA editing. So in order to do this, they screened different types of B3 editors that had various mutations in the ApoB-C1. So the reason that they used mutations in the ApoB-C1 is that we have deduced from this earlier slide that ApoB-C1 is largely responsible for these off-target RNA mutations. So through having different forms of this, they hoped to preserve its function as a deaminase, but also improve its specificity to reduce the RNA off-target effects. So 2 variants were identified that produced very low off-target effects, but also produced high on-target DNA editing. So these 2 are represented in part B here. These 2 are BE3R33A and BE3R33A/K34A. So both of these are mutations in the rat ApoB-C gene. And as you can see, these gray areas are representations of the amount of CTU editing in wild type BE3. And on top, we have in orange the number of mutations that were found from the one variation of the mutated BE3. And on the bottom, we have in blue the other set of mutations from this BE3 with the modified rat ApoB. As we can see in both of these charts, the number of RNA CTU editing across the genome is incredibly lowered. So we also want to make sure that they have the same amount of efficiency when they're trying to edit DNA that's on target. So if we look down here, the targets that they were trying to get to that they decided were great were these HEC site 2 targets. As you can see in the wild type, we've got targeting for the C, and in the BE3R33A, we've got matching targeting for the Cs. So this showed that we have a comparable DNA editing efficiency that's between the wild type BE3 and the mutated BE3s. However, the mutated BE3s were able to produce reduced RNA editing effects. Finally, they decided to look at RNA editing by adenosine base editors. So ABEs introduce A to I alterations when they're looking at RNA. So we went back to liver cells were infected with either ABE or a negative control that was lacking the deaminase. And RNA-seq, again, showed tens of thousands of altered RNA bases with almost all edits being finalized A to G. So again, we can look at the efficiency of DNA A to G editing through this one. So ABE max is the ABE that does include a deaminase section, and the control does not. So if we look at this, we can see a high level of on-target DNA site editing in the ABE max, but not in the control. And then if we look at the part B, we can again see that plasmids that were that organisms that were transformed with plasmids that had only GFP on their plasmid with the control that did not include a deaminase expressed very little RNA A to I editing, while all of the replicates of cells that were transformed with the ABE max, which did contain the deaminase, experienced high levels of RNA ADI editing. And then this is all a replication, again, of what was done with the CBE editors. As we can see through here, a lot of these different diagrams were seen before. But we have similar results in that the RNA ADI editing was fairly uniform across the chromosome. And rather, instead of having the same consensus motif as in the first experiment with the CBEs, in the ABEs, the consensus motif is usually TA and then a fairly even distribution between C, T, A, and G. So this is consistent with the natural target sequence of the deaminase that's used in ABE max. So CBEs and ABEs both induce extensive RNA edits that are inconsistent with DNA edits. And the effects of these unwanted edits will need to be accounted for in research studies. The duration and level of base editor expression for therapeutic applications in humans should try to be minimized in order to reduce the unwanted effects. And because we have these unintended consequences, safety assessments of human therapeutic applications for these type of single base editors may need to include an analysis of the potential consequences from RNA edits. Another important point that they made through this paper is that the creation of secure ApoB-C1-based CBE variants can show that unwanted RNA editing can be reduced without lowering the DNA editing capabilities of CBE. So that opens a window for more research in the future about ways in which these single base editors can be edited to produce greater specificity and less RNA editing. All right. So finally, we're going to talk about a different approach to the way that we can edit our genes. So the paper that we're going to look at for this is Structural Basis of DNA Targeting by a Transposon-Encoded CRISPR-Cas system. All right, so a little bit of background information. Bacteria often use, they often try and take up genes from their environment or from other bacteria using horizontal gene transfer. So there are a number of different ways and mechanisms that they can do this. But two ways that they can do this are CRISPR and transposon systems. So the first thing that we can look at on the top of this page is a classic CRISPR system. So a normal CRISPR system encodes Cas. We're going to look mainly at the enzymes that it encodes. The CRISPR itself, we have talked about in the past, but it encodes a number of spacers and repeats, as well as a guide RNA. And Cas 1 to 3 are used for spacer acquisition. So that would be for incorporating these different DNA targets into the bacterial genome. And Cas 5 through 8 are referred to as the cascade complex. So this cascade complex is used to target the specified DNA sequence that we're looking for. For further use, as we can see, instead of having it go directly 5, 6, 7, 8, these are all out of order. So Cas5 and Cas8 generally form a fusion protein. So they will be referred to just as Cas8 from now on. So whenever I say Cas8, think Cas8, Cas5 fusion. On the bottom of this page, we have a transposon system. So what we have here is TN7. So this is a transposon system that's found in E. coli. And so it encodes 5 TNS genes. That would be TNS A through E. So TNS A and B are enzymes that excise a transposon donor. So that would be a donor DNA sequence. And then they both catalyze DNA integration into bacterial genome. So TNSC is an ATPase that helps provide energy between the TNSAB and these other TNS ones. And then if we look at TNSD and TNSE, These are mutually exclusive targeting factors that specify DNA insertion sites, so where the donor DNA should be placed. So this lab, the Sternberg lab, that wrote the paper that we're going to be talking about for structural analysis of transposon systems, wrote another paper recently about transposons for RNA-guided DNA integration. So for this study, they looked at TN7-like transposons. So TN7-like transposons contain a nuclease-deficient CRISPR-Cas system that can catalyze RNA-guided integration of genetic elements. So if you look at this diagram up at the top, this is the TN7-like transposon system. So we can see that we still have the cascade complex, which is Cas6, Cas7, and here listed as Cas8. Observe that Cas8 is bigger than the other ones, which indicates that it's the fusion of Cas8 and Cas5. So we still have this cascade system. We still have a CRISPR array, which has its number of spacers and repeats, which are used as guide RNA. And then we have TNS A, B, and C. So we still have the TNS A and B that can cut out a donor sequence of DNA and catalyze DNA integration. And then we have TNIQ. So TNIQ is a targeting factor that takes the place of TNSD. So we can see in this Cas system that we're lacking Cas 1 to 3. So we're lacking the part of the CRISPR-Cas system that is able to actually cut out DNA. However, we have instead the ABCTNQ part of the transposon system, which is used in that system to cut out DNA. So by combining these two, we have the means both to cut a donor DNA, to cut an existing DNA strand in order to insert it, and we have through the CRISPR, we have a means of targeting. We have a means of targeting a specific part of the DNA that we would like to insert extra DNA into. So if we look through this diagram, we've got both this TN7-like transposon system on top, along with our transposon itself. So we can see here that the purple target sequence that is targeted by this CRISPR array is bound by TNIQ cascade complex that's formed over here. That would be this funky looking guy right there. And then this complex recruits TNSC in order to be able to use its ATPase, and then TNS A and B are used for integration. So one of the most important things to note here is that we can see that integration of the transposon happens about 49 base pairs after the target sequence. So this shows that this is actually targeting the correct target sequence, and that it has some kind of a specific aim. So that's important because now we realize that this CRISPR-Cas system is integrated and working directly with these TNS enzymes in order to still guide integration of DNA based on a specific target sequence. So now we know that transposon systems and CRISPR systems can work together to integrate DNA based on a guide RNA. So what we don't know is how they work together. We know that this happens somehow, but there are many parts here and we don't really know how they interact. So if we look at, if we remember from the last lecture, cryo-EM uses frozen samples of biomolecules and passes an electron beam over them to generate a model of biomolecules. So this can be used to determine high-resolution structures of biomolecules. So potentially we can use cryo-EM to determine the structure of the transposon-like complex in order to find more about its function. So proteins, the binding and function process of proteins depends a lot on its conformation. So if we're able to determine the structure of a protein, we are able to learn a lot about the way that it functions. So for this purpose, we're going to look at the TNIQ cascade complex and use cryo-EM on this complex to try and figure out what its structure is, and from there deduce what its function is. So the first thing that they did for this study was that they purified the TNIQ cascade complex. So to do this, they introduced plasmids into cells in order to express this TNIQ cascade complex. And if we look at these structures, these represent the cryo-EM maps that were gained from using cryo-EM on the purified TNIQ cascade complexes. So we can see it in a variety of different orientations, and we can both see the naked cryo-EM map and the refined model. So through this, we can see that the TNIQ cascade complex forms a helix. And this helix has two things protruding from either end. It's got one protuberance that is consistent with Cas8, and it has another one that is consistent with TNIQ. The rest of the helix is made up by the rest of the cascade Cas6 through 7 areas. And if we look closely, the CRRNA that needs to bind in order for this system to be targeted to the correct place is located here in gray and is bound to the Cas7 and Cas6 regions. So the natural Cas8-5 fusion in the cascade complex is seen in this C part over here. And the reason that we see it in this chart on the side, but not in this, not in the overall cryo-EM maps and the refined models, is that in order to find it, you needed to have low frequency analysis. So the fusion between the Cas8 and 5, it has a subdomain that's really flexible, and it connects with the TNIQ protuberance, which is right here indicated by the blue and yellow at the opposite end of the complex. And it connects this TNIQ to right here in the middle, as we can see through this gray. So we can't have a really good picture of it in the cryo-EM map because it's very flexible. So that means that its conformation could be pretty different looking at different proteins, and thus when you freeze these proteins for the cryo-EM, it could be in different states, and therefore it's hard to get a really good picture of it. So if you look at the structure of Cas7, we can see that Cas7 protects the CRRNA by forming a helix along its length. So you can see it a little bit better in this refined model down here, where this gray snake that's going through the center of the cascade complex is shown to be almost completely protected by this Cas7 portion of the complex. So Cas7 is rather large, and it's been divided into 6 different sections. So the way that this works is that the Cas7 kind of surrounds the CRRNA in a helix, and it clamps down the CRRNA at regular intervals. So every 6th nucleotide is flipped in the opposite direction that it's supposed to. So this causes it to be more stable, but also leaves the rest of the CRRNA available for DNA recognition, because that's going to be important when we're trying to find a DNA sequence to bind to. So Cas6 and Cas7.1, which you can see down here at the base of the CRRNA in the red and the green right at the base, interacts to form the 3' CRRNA handle. So as we can see, there's the end of the CRRNA interacting with the Cas6 and the Cas7.1. And in order to do this, an arginine-rich alpha helix is positioned in a groove within the complex that positions basic residues, which are positively charged, within interaction distance of the negatively charged RNA backbone of the CRRNA. So this allows the CRRNA to be bound with the Cas7.1 and Cas6. So the Cas6-7 interface forms a surface that can dock TNIQ. So it's found that TNIQ binds to cascade in a dimer with a head-to-tail configuration. So we can see this over on this side, where both the blue and the yellow are TNIQ. TNIQ has two separate domains. It has one domain, which in this diagram is labeled the TNIQ domain. And that domain is comprised of alpha helixes and has the normal structure for any proteins in the TNIQ family. And then its second domain is the N-terminal domain. And this takes the shape of something called a head-to-head domain. So a head-to-head domain is usually used for nucleotide binding or nucleic acid recognition, but it can also be repurposed for protein-to-protein interactions. So the head-to-head proteins that are indicated on each part of the dimer help to bind the TNIQ molecules together. So as we can see here, the alpha helixes of H3 and H12 are bound on this end of the domain, and the H12 and H3 are also bound on this side. So these are lined up asymmetrically. They're lined up head to tail with the C terminal of one protein aligned with the N terminal of another, and vice versa on the other side. So in this way, we can see the structure that the TNIQ takes when it's forming a dimer to be bound to the cascade complex. So we can also see on this diagram, it's indicated where Cas6 and Cas7.1 can bind the TNIQ domain. So a better picture of this is on the right-hand side, where we can see that the TNIQ1, which is represented by the blue, in its dimer with the TNIQ2, which is represented by the yellow, we can see that it fits into the Cas6 and Cas7.1 that are holding the CRRNA. And the reason that it does this is that there are different hydrophobic regions in the Cas6 and the Cas7.1 structures that allow the TNIQs to bind to them at specific points. So HTH is used, this portion, HTH, is used mainly for the dimerization of TNIQ, and the TNIQ domain of the TNIQ are the ones that are generally used for binding to the cascade complex. Finally, they looked at the DNA-bound structure of the TNIQ cascade complex in order to try and determine what kind of structural aspects of the DNA are needed for recognition by this complex. So in order to do this, they looked at the complex when it was bound to a double-stranded DNA that had a 32 base pair target sequence. So in this case, we can see that Cas8, right here in the purple, docks where there's a PAM. So a PAM is just a short stretch of nucleotides. It's only a few long. That helps indicate a normal CRISPR Cas complex system where it should bind to a target strand. So we can see here that we have in red, we've got our target strand. It's being separated right now because it's got this gray CRRNA, which is binding to its target sequence. And we've got, looking off into the right-hand side, we have the former complementary sequence of the DNA that we're looking to edit. So this again can show that all parts of Cas7 are linked to the length of the CRRNA, which is helpful in binding the target sequence. And then we look at the Cas6 and TNIQ dimer down at the bottom. If we look at part B, we can see these are the cryo-EM generated pictures of what the structure of the TNIQ cascade complex might look like. And for a closer look, these were developed into models that have, in the light gray, we can see the cascade complex itself. In the dark gray, we can see the CRRNA or the guide RNA. And then in the red, we can see the DNA that's bound to it. So Cas8, as we can see, docks the PAM, and the CRRNA is unwound where base pairing and CRRNA begins. So the 22 nucleotides of the target showed clear density, which means we can determine which nucleotides these are and their order. However, 9 of the last nucleotides were not ordered. So this means that there was no observed density for the target strand nucleotides that bound to the CRRNA by Cas 7.2 and 7.1, which are down over here. So this region is supposed to be flexible owing to the interactions of the Cas8 flexible domain with the TNIQ dimer as shown above. But these disordered nucleotides also correspond to an area of the target where RNA and DNA mismatches are detrimental for RNA-guided DNA integration. So therefore, we can see that this thing on the bottom, this little guy right here, is called an R loop. So an R loop consists of the CRRNA guide RNA that is bound to its target sequence. And then there's also this loop that's formed where there is space now in between this formed double-stranded DNA RNA binding and the existing complementary sequence of the target. So the because the disordered nucleotides that they couldn't figure out a specific structure that was needed correspond to this end part of the R loop over here, where RNA DNA mismatches are detrimental for the RNA-guided DNA integration. They proposed that this partial R loop structure could represent an intermediate conformation, and that further structural rearrangements might be critical for stabilization of an open conformation, which would be over here, the R loop locking. So the cryo-EM structures of the CRISPR-Cas effector complex bound with TNIQ, both with and without DNA attached, were analyzed through this experiment. And the structures revealed the presence of TNIQ as a dimer that interacts with Cas6 and 7 in the cascade complex. This structure is probably a recruitment platform for downstream acting transposition proteins, and there's possibly a fidelity checkpoint where formation of a complete R loop requires conformational rearrangement that could depend on extensive RNA-DNA complementarity or downstream factor recruitment. So this could account for the highly specific RNA-guided DNA integration that was previously reported for the TN7-like transposon. So through this study, where we tried to figure out the exact structure of the TNIQ cascade complex with cryo-EM, we were able to deduce a little bit more about the possible functions of this complex as it works together, and we're able to more completely flesh out the possible mechanisms for its workings as were discovered in the paper before.
So thank you for listening to our lesson today. So just to recap, we went over the 2 very broad categories of how pure science projects are designed versus how engineering projects are designed, and we went through examples of both of those designs for a more concrete idea. With pure science, largely your results drive the future experiments that you do. For engineering experiments, typically there's a step-by-step process in which you are always trying to reach the final goal of creating what it is that you want to create, and each step gets you one step closer to that goal. And it's usually it's usually predetermined a bit more than pure science projects. So then we brought you through trying to predict roadblocks in scientific design, particularly when theory and reality are not quite the same. First, we looked at the Trojan horse cheater model and saw that when the theory relied on the fact that every single cell will be eliminated, the reality is that in biology, typically you cannot eliminate every single cell, and therefore this method, that would be a roadblock, and this would be a difficulty in having this be successful when applied. But there are also roadblocks that you just can't predict, such as the base editing system, where you're not going to know how severe the off-target effects are until you build the system and then you test the off-target effects. And we see that there are pretty substantial off-target effects for base editing. The base editors, but at the same time, there is the possibility for optimization of these systems. So it really depends on whether or not you can get an optimization of the base editors that were described, in which the off-target effects will be negligible. And finally, as you see with the base editor example, while one group is working on base editors in one way, other groups could be working on genome editing in another way. You see the work from the Sternberg lab combining the power of transposons with the CRISPR system and the CRISPR-Cas system, and using that method, then you have integration. So just to recap, integration is when you have the insertion of a particular DNA sequence into the genome of an organism. And you might notice that in this example with the transposon system, you will be inserting a decent amount of bases. So it's still a large number of bases that are being edited. And in this sense, like, it's still different from the base editing, but there's ongoing research going on in parallel on alternatives to genome editing, et cetera, et cetera. So when you think about like what type of research project would be best to do, think about not only how one tool can be applied, but think about whether or not is this the best approach to apply to the problem that you want to approach. So with this lesson, as you can probably see, science is hard. That's why we have to train for so long in order to fully become independent researchers, and why you go through so many years of schools to finally be able to lead your own laboratory. But hopefully this does not deter you from science, in that although it's hard, things that you can do are honestly pretty remarkable. In that now we can do some genome editing, although there is some optimization that needs to be done. We have come a long way, and we've gotten a lot closer to developing a system in which we can really accurately edit the genome. So in our next lesson, we're going to talk more about the key factors to good proposals for scientific projects. We're going to talk a little bit about the largest National Institute of Health grant, and we'll talk more about the bioethics of scientific projects, which we should always consider when designing our projects. So thank you for your attention, and we will see you next lesson.